Well, I don't want to disrupt your eating, but I do want to say a few. I do have a few announcements. This is also our annual business meeting. I would like to welcome, for people who have who have come for their first luncheon, Dave Deemer, June Gordon, David Sweet, Bill Hyde, and David Dorfan. And special thanks go to David Dorfan. You'll never guess why. He is our 170th member, and it means that we have added 20 new members this year on top of the 50 members that we added last year. So that's 200% of my goal of adding 10. And so that made me very happy, and I told him about this, and he's overjoyed. Wanted a discount. I'd also like to thank our officers who have worked double duty for this strike event. Our caterer said they didn't know whether they could serve the food today. They would know this morning. And I said, how do I process that? So Don Massaro, Christy Dolly, Virginia Jansen, and her associate Melody Corcos, Virginia Rivera, and for helping out and covering also for officers that are away and traveling. I'd also like to thank Noah Sullivan, who's a first-year student that is our AV assistant today. Give him a big hand. I'm hoping we can recruit him and have him be here every luncheon until he's a senior and becoming a graduate student. So, his background is he was the AV person for his high school or college yearbook, high school, I guess, for his church or something. I mean, this is what he's done. He's taken classes on it. So we have arranged for this Arboretum for the next 3 years. That's the good news. The bad news is they doubled our rent. But no negotiation was possible. So what we saved on this luncheon will help allow us to hold the luncheon price fixed for this year, and the dues as well. Greta is on the East Coast, so she is being Dom Massaro is taking over for her today, and you saw that. Today we have a business meeting. This is going to be ultra short. I'd like to ask Barry Bowman to step out briefly. Barry is vice president today and is up for promotion to president. He's been understudying and has passing grades. I'd like to ask if there are any other nominations for the position of president. Seeing none, I'd like a show of hands for those in favor of Barry serving as president, starting July 1st. Okay, any opposed? Any abstentions? Barry, could you come back in? So let's join me in welcoming our future president. Barry starts the 1st of July, and I'll become past president. That leaves the position of vice president vacant. So we're welcoming, and not necessarily today, that we have volunteers or nominations for somebody to be our program chairman for the next 2 years and move then to the position of president. And during that time, there's training and so on. The other positions that we're seeking nominations for is the position of secretary. Virginia Jansen is serving that role, but would like for somebody else to take her position. She'll still continue to send condolences to those that pass away. And the position of event coordinator, Al Smith has been in this role for some time. He's happy to continue, but he would also be even happier if somebody else who didn't travel as much as he does or could help out or take over. Okay, so that is the end of our business meeting. Normally we would have changes to our officer's handbook, which is our bylaws. Why is this? But they're not ready yet. So those will be circulated later, basically defining and updating the positions as the duties have changed. And so that is the end of our business meeting. And now this brings me to a special time. You're aware that we have an endowment. I wrote about it in the last newsletter and have mentioned it at a previous luncheon. But I didn't want to disclose the donors, until they could be here with us from their home in Palo Alto. He is a professor emeritus of mathematics. He taught from 66 to 94. She is a former lecturer. He was UCSC's first associate academic vice chancellor for undergraduate affairs and was provost and senior preceptor for academic affairs at Crown College. They've also created a fund. Can you turn it down a little bit? Created a fund for future teachers of math and science. He has been a pioneer and leader in math education statewide and nationally. Now, it's my great pleasure to announce the Edward and Miriam Landesman Emeriti Fund and their gift of $50,000. I'd like to ask Ed if you'd like to say a few words.
Sure. Well, thanks, Todd, and it's really great to be here. We don't get over here that often, but it's great to be here and see so many people that I know and haven't seen in quite a while. I'd like to say that Miriam and I began thinking about an endowment about a year ago, and we saw a number of different projects that were worthy and that could help the campus, but we kept being drawn back to what the Emeriti Association has been doing under the leadership of Todd Whipke. And it didn't take too long for us to say, that's what we want to fund. And in many ways, we see the Emeriti Association taking care of us in ways that maybe we were taken care of when we were regular faculty teaching and doing our research. But I'd like to say a few words about Todd. He's only given me 5 minutes, and that is no justice to what he's done, but I'll stay with the 5 minutes, maybe 6. As president of the Emeriti Association for the last 2 years, he's met and interacted with the Council of UC Emeriti Associations, the Council of UC Retiree Associations, the System-Wide Faculty Welfare Committee, and the UC Retirement System Advisory Committee. In many ways, I see the Emeriti Association as the checks and balances for what could be done relative outside of here, and he's done that. And I made a list, and I'm not going to do justice to him, but I made a list of the things that he's done, and it's really worthy of mention. One, as some of you may know, 3 members in UC had their pension checks stolen, one from this campus. He took on the lead on that and made sure that those people got back their pension payments, and also at the same time made at your service, online, a much more secure place with regards to their whole security system, that was a task that was unbelievable. He also fought the right for retirees to sign up for Kaiser Permanente here in Santa Cruz. Another task that would not have been done without Todd getting in there and really kicking. A third, he made public the loss of emeritus status of one of our members, and requested that privilege and tenure hearing for that person, and that's been granted, and in fact the status has now been given back to that person, at least until he gets through that privilege and tenure committee. 4, he publicized the travesty that befell the Science and Engineering Library and the lack of shared governance of UCSC regarding the library. Another big task. 5, we now have, the Emeriti Association now has presence in social media, Facebook, YouTube, Twitter, Instagram, a YouTube channel. Thousands have now seen what's on that relative to what we do with regards to the research that faculty as Emeriti do, with regards to their teaching and service that they do. The Luncheon talks have produced videos, and he now has that set up in a way that people can look at those videos at any time, whether it be today or years later. All the work that he's done, it's hours and hours and hours, and that's what he's done as chair. I've got a few more things, and I'll leave out some, I'm sure, and I apologize that I'd leave out some. There were 4 special events in addition to the talks that are given here. One of them was one that I was able to attend, the Crown Emeriti. Let's see if I have that here. Yeah, it was the Crown Emeriti Luncheon. He's had 2 sailing events for us. One will be coming up this July. And also on the impact of the tax cut law, had a get-together on that. The newsletter that you're reading that is put out now, due to Todd Whipke. There was never a newsletter 2 years ago. And now that newsletter really brings us together in a way that we're able to see what our colleagues are doing with regards to research, teaching, and service. Just superb. The newsletter's brought us 29 articles to date. Maybe as of today it's even more. And finally, the last one, to show the detail of the way he does things, I had an email yesterday saying that we should try to carpool, and it said that I should try to carpool with the person that lives near me, and they mentioned my wife. Now, so they were arguing, in mathematical terms, they were arguing she was less than epsilon distance from me. This is the one time when epsilon is 0. We live together. I think all of us need to give Todd Whipke an incredible thank you for what he's done for this organization.
Thank you, Ed and Miriam, on behalf of the Emeriti Association and all Emeriti Associations members to the infinite future, because this endowment is a permanent benefit to our association forever and ever, beyond our lifetime and our grandchildren. So it's really remarkable, and I want to thank you. And thank you for the kind words that you said about me. Thank you. So, Barry, I'd like you to come up and introduce our speaker, and then the speaker will speak, and we'll have questions both during and after, I guess, the talk, and then that'll be the end, except for picking up more pizza.
Okay, don't go away quite yet. So just to add to the great list that was given here, there's one other thing that Todd has done that may have taken more time than anything else. How many of you have gotten a message from Emma? You're wrong. Every one of you have gotten a... How many of you know what Emma is? Just a few. Todd created Emma. Emma is our software robot who really runs this entire organization. You wonder how persistent the messages can be? That's Emma. All right, it's a long message. But we thought, as the officers of the organization, we should give Todd some kind of permanent thing to show that we really appreciated what he did. And so we came up with this, not too tacky, kind of, you put it in the window and it lights up. This is an appreciation for outstanding service, Todd Whipke, President of the Emeriti Association, from 2008 to 2010 and 2017 to 2019. He's done it twice. So thank you very much, Todd. Okay, so today's speaker, let me get this right here. Today's speaker is Carrie Partch from the Department of Chemistry and Biochemistry. Carrie was an undergrad at the University of Washington, and she's really gone all around the country in her education. From Washington, she got her PhD at the University of North Carolina, and then she was a postdoc at the University of Texas Southwestern, and then finally came to Santa Cruz in 2011. She works in this fascinating biological question, and I think she's one of our more popular speakers on campus. And she's a superb teacher and has done superbly well as a really new professor. She's won the major award from her scientific society, and she's going to tell us today about how our biological clocks really work. Carrie does this great combination of kind of whole human animal physiology behavior while also doing a detailed analysis of the structure of the proteins behind this clock that is in every one of our cells. Carrie.
Thank you. I'm going to go ahead and get set up here. Is this on, that part? Okay. Okay, I cannot get that thing pinched together.
Okay, yeah, just look on there. Okay.
Okay, and it was just kind of hard to pinch. Okay, yeah, it's really a pleasure to be here today. Oh, sorry, this is falling apart. So I want to thank Barry for the introduction and the opportunity to speak. Can everybody hear me, even in the back? Yeah? Okay. It's really a true pleasure to be in front of my esteemed colleagues and guests, and it's really an honor to be able to talk about what we do in my lab and our sort of interest in biological timekeeping, which I think is just a fascinating area because it really spans beyond the human experience. We've all probably experienced jet lag, right? I'm guessing we all have. And as you'll see today, our understanding of circadian rhythms underlies that. And I'll give a note that I'm happy to take questions during the talk if any arise, or take them at the end. So however you guys want to roll with this. But what I'd like to do is really focus on sort of the breadth of this phenomenon on Earth. And so I have a picture here to sort of remind me to show you that as all life evolved on Earth, it really was faced with a persistent and consistent and pervasive environmental change. And that is that as the Earth rotates about its axis, we're exposed to routine cycles of night and day. And nearly all forms of life on Earth have evolved biological clocks within them to measure out this 24-hour timekeeping within themselves and use this to anticipate the coming of the next day. So I'm showing here at the top several photosynthetic species from single-celled cyanobacteria all the way up through these beautiful sunflowers. And of course, as a photosynthetic organism, these particular species have a very intimate and dependent relationship with sunlight because it's their food, right? And so these early forms of life, including cyanobacteria, evolved a way to anticipate the coming of the next day. So I want to show you a movie that I think truly encapsulates, to me, the wonder and amazement of circadian rhythms. And what we're looking at here is a baby sunflower. And what it's doing right now, it's not a very exciting part of the movie, but it's tracking sunlight throughout the day. Right? It's tracking its food. And as we approach towards the afternoon and dusk, the plant here, as you can see, goes to sleep. And I'm using that word very loosely. But here's where you can see the beauty of circadian rhythms. This plant knows that the morning is coming. It wakes up and it points towards the east. So it's ready for those first few delicious photons that come in the morning. And it's this ability to anticipate the coming of the next day, right, to anticipate a regular environmental change, in this case, its food source, that is the reason we think circadian rhythms arose in life on Earth, right? It's important to know when your food is coming next, right? And so this isn't true just of photosynthetic organisms, but in fact, nearly all forms of life on Earth, from insects, invertebrates, and on up to people, have the ability internally to tell 24-hour time, and we use it to coordinate the physiology within our bodies and our behavioral responses, right? Okay, so I wanted to walk you through, I know that I apologize for small text here. I wanted to walk you through some of the many different behavioral and physiological responses that are controlled in your body by circadian rhythms, because I could spend the rest of the afternoon going through a list of them. But what we know of, so what I've plotted here is sort of from 2 a.m. all the way through the day, and it happens in the midpoint of the night, the midpoint of your sleep. Your body reaches its lowest body temperature, right? And this is associated with a nice deep sleep. That usually sets the timing that you wake up in the morning, and I'll come back to this later in terms of how the circadian rhythm interfaces with the timing of sleep onset and waking, sort of the theme of the talk today. What we do know is that right after you wake up is your normal time of highest blood pressure, right? And so this happens to be the time that most people think of as being associated with dying of natural causes. And so there is a circadian rhythm of sort of the normal onset of death, and it's because of this spike in blood pressure that happens normally every morning, right? And so as we move from sort of early morning to mid-morning, we know that, in fact, you have your peak ability to concentrate and your highest cognitive performance midday between 10 a.m. to noon. So I like to joke to my students when I'm teaching in the morning that I get them literally at their best, right? And so there's sort of a peak in your awareness that happens throughout the day. Right now, if you wanted to enjoy some coffee, it's actually the best time, because if you've ever had that post-lunch dip in attention, it's not because you've eaten, it's because the circadian rhythm has a lull in promoting alertness and wakefulness, right? So this is the best time to have a cup of coffee if you want to promote that, right? And likewise, it turns out that the circadian rhythm can actually predict the best time for you to work out. That later in the afternoon, you have sort of your best pain threshold and your highest ability to add muscle mass. So if you want to get the most bang for your buck, working out in late afternoon is the best. right? So these are kind of fun behavioral responses, but we also know in work that originated from my graduate lab at UNC that actually there are molecular consequences to circadian rhythms as well. So for example, if you go out and were to take a walk in the arboretum, we know that exposure to sunlight induces DNA damage in your cells, a shocking amount, 10,000 incidences of damage per cell per day that need to be repaired to maintain your integrity of your genome. And so you have vast protection systems in your cells that do that, and it turns out the peak activity of that process happens at the end of a day. So as you've accumulated DNA damage all throughout the day from sunlight and other exogenous sources, there's a peak in that activity that happens in the evening. And finally, one of the most obvious things and exciting things, I think, as we'll come to in the talk, that the clock controls is the timing of sleep onset. In other words, when you like to fall to sleep. And so this is largely controlled by the hormone melatonin. I'm sure everyone here has heard of melatonin, and can I get a show? Does anybody routinely take melatonin, like daily or when they travel? So it has sort of variable efficacy because it's not clear to know when you should take it. So what I've plotted in this blue line is actually the rhythm of melatonin expression, or the abundance of melatonin. So during the day, melatonin is low, and as the sun goes down, melatonin levels start to come up and help to prepare you for sort of your sleep onset. And so this is a circadian controlled process so that it's low during the day and high at night, right? So that curve, I'll go back a sec. This curve is a little hard to see because the 24-hour day is kind of split, right? But if we were to measure the amount of melatonin going from mid-afternoon to mid-afternoon, you can see that there's a nice peak of expression of melatonin that happens in the night. And this is what we call a circadian rhythm, right? That something happens once per day. There's sort of a peak of activity. And it turns out it's not just the genes that make melatonin that are under circadian rhythms, but nearly 10,000 genes in your body undergo a change like this, where the peak protein that's made is made at one time of day. And by controlling the rate-limiting enzyme for a process, that's how a circadian rhythm can control when that process will happen in your body. So for example, I mentioned DNA repair. One of the key factors that recognizes damaged DNA and sits down on that particular site is under control of the clock, so it's only made in the late afternoon, right? So I think as our understanding of circadian rhythms grows, there's been a really exciting connection that I think has very real life implications. And I realize no one likes to look at a Venn diagram, but what I'm showing here, if we take sort of modern knowledge of genes in our body that are associated with many forms of disease, from mental health to metabolic disorder to cancer, this is the sort of tan circle here. So there are thousands of genes that have been implicated in human disease. And if we look at those genes that are under control of circadian rhythms, the sort of 10,000 or so genes in your body that exhibit this beautiful pattern of being low at one time of day and high at another, there's an overlap of thousands of genes. In other words, there is a time component to controlling the expression of genes that participate in processes that can go awry in disease. And I think the most exciting aspect of our field is to harness this power to improve therapeutic treatment of disease. So for example, of the known FDA-approved drug targets, there's about 14 or 1,500 or so. There's nearly 1,000 of them, of targets of the, if you take a pill for a drug, that 1,000 of those are under control by circadian rhythms. And there's really a growing appreciation that the timing with which you take your medicine might actually help reduce drug dosage, improve efficacy, and remove or reduce off-target effects. So this growing area of the field is called chronotherapy or time therapy, and it's really, I think, beginning to take hold. And there's been a huge appreciation not only for when you should take medicine, including chemotherapy. We know that there are times of day we can give drug doses, excuse me, that reduce off-target effects and enhance efficacy, right? So there's just, I think, an immense balance to appreciate the effect that time has on biology, right? So I want to walk you through how it is that we came to know what circadian rhythms are and their importance, right? This is sort of a very modern view of understanding this molecular clock in your body. And really, we have to go several hundred years back to understand the sort of initial scientific inquiry. So there were French philosophers that had observed plants doing exactly what I showed you, going up during the day and down at night. And so there's a very famous French philosopher who first stuck a plant into a closet and removed it from sunlight and saw that it still went down at night and it still came up in the morning, even in the absence of light, right? So we had human endeavors for hundreds of years have understood that there was something internal in organisms that could measure out about 24 hours, right? And this is the sort of genesis of the name circadian, is that it's an about a day process that happens internally. But it wasn't really until the 1960s where we had sort of modern research that began to inquire about human circadian rhythms. And this was largely the work of Jürgen Aschoff. And so just like that French philosopher who stuck plants in a closet, he stuck people in a World War II bunker. And he completely removed them from the sort of temporal cues that we get from the normal 24-hour light cycle, right? So to prove that this isn't just merely a response to the sun, but in fact something that's encoded within us. And so this is a picture of this World War II bunker, this very famous, scientifically famous bunker that he used. And what he would do would take patients, so over about 30 or 40 years, he had thousands of people coming in, over 400 studies, in which he would put them in what we call temporal isolation. So they weren't locked in complete darkness, but they were no longer aware of what the external light-dark cue was, right? And then he measured body temperature and sort of easy, physiologically easy things to measure. So he measured urine excretion. Turns out that the volume of urine that you produce over the day is controlled by the circadian clock. It's low at night, which I'm thankful for as the mother of young children, that that changes at night, right? So here, for example, is a photo of one of the study participants, coming out and greeting the sun after 10 days in temporal isolation. And so I want to show you a figure from his really, I think, seminal paper in 1965. Yes?
How long did people stay there? You just mentioned 10 days.
Yeah.
And then was they there a long period to perhaps readjust the clock?
That's a fantastic study. So his studies tended to run between 10 days and 2 weeks. But there are many other studies, including some that happened before him. So Nathaniel Kleitman, he was one of the co-discoverers of rapid eye movement, or REM sleep, a preeminent researcher in the sleep field. Back in the 30s, he went into the Mammoth Caves in Kentucky and put himself in temporal isolation for a month to try to adjust to a 28-hour day, which we now know to be a very sophisticated scientific experiment. Right? So there are people who, throughout the last 60 years, have put themselves in isolation up to 2 to 3 months to try to get at this question. And I'll tell you now, NASA is working with recruits, I would say, or volunteers, to put them into isolation up to 500 days. Right? And part of that is to see if they can adjust to a Martian day, which is 25 hours. So I'll come back to that at the end. Right? So it's definitely an ongoing area. And so I'm going to show you this figure because it's, and I reread this paper actually this morning, and it's a really amazing, elegant read. I'd be happy to email it to anybody if they want. And so what he's showing here, each of these sort of vertical lines is showing you a 24-hour terrestrial day. And I'll highlight one of those days, and hopefully you can see that there's a sort of undulation. This third column down here, volume, sorry to be a little indelicate, I know you're eating, is urine volume. And you can see that it goes up during their day and it comes down at night. And then he's also measuring the calcium and potassium that are excreted, right, as a function of that. And down below, he was measuring body temperature. And hopefully what you can see is that there's this beautiful undulation of going up during the day and down at night. And this happened on a 24-hour rhythm, completely independently of the terrestrial world outside. These subjects would get up when they wanted, they'd turn the lights on, they'd go to bed, because our body runs on a 24-hour rhythm internally. Yes, in the back.
We know these changes if you move to Europe, you accommodate pretty quickly. Is there a fixed number or pretty close to a fixed number, or is it some people do it 2 days, some people take
Yeah, so this is a great question. So light is one thing we'll come around to a little bit is that light is a really powerful drug for our system. The way that these circadian rhythms work is that internally we have a rhythm of about 24 hours. We'll touch on that a little bit later, but that it gets locked to the terrestrial Earth with light exposure. Right? So light exposure through our eyes going into what we call the master clock in our brain communicates when the day begins and when the day ends. So when you fly from here to Rome, for example, and you have a 9-hour shift, you have to adjust for that 9-hour shift in your internal timing. And it works out that it takes about an hour. For every hour you travel in a time zone, it takes about a day to catch up. right? And so many of us in the field are interested in studying this process to understand how it is that this light information is communicated to our clock so that we could eventually try in ways to regulate and control that, right? So that's a pretty typical shift. Some people adjust faster, so they shift in their circadian rhythms faster than an hour, 1 day per hour. We call them super shifters, and we're studying them to understand why that happens. Yeah?
How about the intensity of the light?
Oh, intensity of the light. So our circadian system is exquisitely sensitive to light. So even moonlight, low light. And in fact, this is getting me off topic from circadian rhythms per se, but there are really dramatic ecological consequences to light at night. So, because light communicates daytime to our bodies, so when we turn lights on at night, what we're doing is pushing our circadian rhythms back, right? We're sort of telling our bodies it's still daytime. And I'll tell you, our bodies do different things during the day than they do at night, right? And so not only do those consequences happen for us, but also for the terrestrial environment. And so I saw an amazing talk recently about sort of ecological consequences of street lights and the effect that animals that use moonlight no longer see moonlight because all they see is street light intensity. So we're very sensitive to light. So by the way, I have blackout curtains in my bedroom, right? Yeah, and you should too. Yes.
So your volume thing up there moves quite a bit during day to day. So how constant is this?
So what I would say, to answer that, I would say that you can see that it's kind of very pixelated and binned. And actually, I got this from the paper that I read and that I reread it today. The volunteers in these studies were collecting their urine at their own discretion. And so it's not the cleanest measurement. Clean's maybe not the right word to use. But now we have much better molecular tools, and I'll show you an example of some of this. It's really a stunning rhythm. Right? So this was, I think, the beginnings of the molecular era of circadian rhythms to understand that deeply within our cells and our bodies, we have the ability to tell 24-hour time. Right? So I want to just briefly touch on the fact that in our field, one of the most exciting advances recently was that in 2017, 3 researchers in our field received the Nobel Prize for physiology or medicine, as this is the quote from the Nobel Committee, for their discoveries of molecular mechanisms controlling the circadian rhythm. Right? So as scientists, once we understand that these rhythms occur, I think the field really went in 2 areas. The first was to understand what are all the things that are controlled by circadian rhythms, behavior, physiology, and how does that occur? And then sort of following in lag step was understanding what is the molecular basis? How is it that in our genome we encode the ability to know what a 24-hour period is? Right? And so those 2 sort of research goals have really driven the field for the last 30 years. So I want to walk you through the fundamental studies that led us to understand what a circadian rhythm is, and then we'll talk a little bit about how that changes across the human population. So really the story didn't begin with these 3 gentlemen, but in fact these 2 here. So are Seymour Benzer and Ron Konopka. So this was taken many years after his PhD studies with Seymour. But Seymour was really curious to identify, are there genes, can you identify a single gene that controls behavior in some way? And this was somewhat of a heretical statement back in the late 60s and early 70s. And what they did was use this model organism. So this is the Drosophila melanogaster, or fruit fly. And they used it to try and identify genes that might control behavior. And it turns out that one of the behaviors they initially identified that might be amenable to this analysis was a circadian rhythm. And it turns out, so I apologize, I know this is really small. This is a plot of these little pupa emerging from their cases. So they basically are born during the day. Fruit flies, come out of their pupal casings at a particular time of the day. And if you ask sort of a normal animal, the gray bars here are what would be daytime, and then the black bars are nighttime. And if you measure the rhythms of about how long this rhythm occurs, it takes about 24 hours. And so this was the basis for the genetic screen they did. They mutagenized these flies and then looked for flies that had a different behavior. So they found 3 types of flies. This is, again, I think one of the most fundamental groundbreaking studies in our field. They found some flies that completely lost this rhythm. The clock internally had been broken. It was arrhythmic. They found some flies that you can see here did 4 cycles, that still had a rhythm of being born, I'll say. But here you can see that they've accomplished those 4 cycles in a much shorter period of time. And in fact, if you measure out the period or the timekeeping that takes, it they have a short clock. It happens with about a 19-hour timing. And likewise, they found some that in that same period of time only did 3 cycles. In other words, they had a longer clock. So out of one experiment, they found all of these genes that either broke the clock, made it short, or made it long. And they did the painstaking genetics to track it back to a single gene on the X chromosome. And they named this gene period because it seemed to play an important role in setting the timing of the circadian rhythm. All right, so that was 1971. That paper was cited 8 times in the first decade it had been published. It sort of sat there unnoticed, right? And I think largely because the techniques and tools that we needed to identify those genes and clone those genes were not really in common use yet. But as you move into the 80s, a number of people, including these 3 gentlemen shown here, so Michael Rosbash and Jeffrey Hall were at Brandeis University. Michael Young was at Rockefeller. They were engaged in a scientific death match, right? A competitive match to figure out, could they identify the gene, what was the period gene, and in fact, what did it do? And I'll tell you, they succeeded, right? And so I'm just sort of representing DNA here schematically to identify that this is the gene period. That gene is eventually made into a protein we call period per for short, right? So over the course of maybe 15 years, they identified the gene and actually mapped the coding sequence of the gene. They identified the other 2 proteins, which were cleverly named clock and cycle, that actually made that gene from DNA into RNA and then into a protein. They identified a protein called timeless, or TIM, that when you don't have timeless, you don't have a clock. So I don't know if you know, but Drosophila researchers are famous for having funny names for their genes, right? And then the key thing was that they put all this together into what we call a genetic network that explained how the clock gives rise to 24-hour timing. So that's kind of the picture that I'm showing here. So the idea is, if we were to measure the expression of the period gene, during the day, these 2 proteins are making that protein accumulate. And that's what we're looking at with this this gray line, rather. What happens is, as these proteins accumulate, this sort of bar symbol is that these proteins work to turn off clock and cycle, right? This is called a feedback loop. So the levels of the per protein go back down. And when the levels of per aren't there, these turn back on. And so you get this rhythm where it goes up during the day and down at night, up in the day and down at night. And it's this oscillation in gene expression that happens with a periodicity of 24 hours. So the time from one peak to the next is the timing that is set and encoded by your body. And so what we see here doesn't just happen for period in terms of the core clock gene, but now we know that it happens for nearly 10,000 genes in your body. And it results from this sort of network that we see here. So it was for this discovery, identifying these genes and how they fit together into this network, that they were awarded the Nobel Prize in 2017. Now we know that in mammals and in humans in particular, that network is still the same. The names are a little bit different. Some of the genes are slightly different, but it works exactly the same way. So this is a fundamental way of keeping track of biological timekeeping from cyanobacteria all the way up to humans. And I want to show you this actual clock ticking in an individual cell. So what we can do is, instead of looking at expression of these core clock genes, as we would call them, we can hijack a gene from the firefly. So I don't know if you've heard of the gene luciferase. It's the gene that gives rise to the beautiful glow that fireflies have. We can wire that into this network so we can watch the cells glow, right? And as they glow, that's the clock turning on, or expressing the gene. So hopefully you can see here. I know it's really dim. Do you see maybe things twinkling? It's kind of bright in here. So each of these little specks is an individual cell. And the point here that I'd like to make is that this timekeeping happens at the cell autonomous level. It happens within every single cell in your body. So much like Carl Sagan would talk about billions of stars, you have billions of clocks in your body. Every single cell has a circadian rhythm, and at a tissue level, they work together to control the timing and physiology of what that tissue is doing, right? So I don't know if you can, the box is really tiny, but right here in the middle, where this little arrow is pointing, sorry, this is not so great, but hopefully you can see that this is actually reading out the photons that are being emitted, the light that's being emitted. And do you see that it's going up and down every day? It's happening at every single cell. So this is an example of a single cell. If we look at this process in an intact tissue, so this is what we call the master clock in your brain and your hypothalamus. It's wired to your retina. the suprachiasmatic nucleus. It's a fun thing to say. You can see that there's this beautiful rhythm as these genes go up and down each day. So this is happening in every single one of your cells to control what happens at each time of day. So this is really how I fell in love with circadian rhythms as a graduate student. I love how proteins communicate with one another and convey information. And for something to happen so regularly and robustly that it makes a clock was fascinating to me, right? To try to figure out how does this work. Okay, so our clock is, I've added a few genes in here. We have some redundancy. So instead of having one per protein, we have two. Instead of having one of the sort of timeless homologs, we have two. And so these together help to make our clocks really strong and robust. And I want to tell you that we've gone a long way since the 1960s in terms of our ability to measure circadian rhythms. Now with molecular tools, we know at the human population level that as sort of every species has sort of a set point of what their typical circadian timing is. As humans, ours runs just a little over a day. So 24 hours, 11 minutes, give or take a few minutes. That's how long all of your clocks typically run. And if you were sort of in the average cohort, that would give you a bedtime onset of around midnight. And so what I want you to do is instead of thinking these as circles or as proteins, they really are molecular cogs in a biological clock. And so as we all know, sometimes clocks run slow, right? I was just resetting my microwave clock this morning, right? And so when that happens, what are the root causes of that? And I'll tell you, we now know that there are changes in the gene in these very genes that affect how these cogs interact with one another that then read out in changes in biological timing. And that's what I'll spend the last maybe 15 minutes of my talk going over. We now know that if your clock runs shorter than this 24 hours or so, you tend to be a morning lark. People who love to get up early in the morning but don't love to stay up so late at night. And likewise, we now know that if your clock runs longer than this, you are a night owl. And this is largely due to the circadian control of melatonin. Right? So when melatonin starts to come up later in your body, you're not sleepy until later, right? But if your clock is a little shorter, melatonin starts coming up 6 or 7, you're getting sleepy before your friends maybe, right? And so to me, this was a really fascinating tool. And as I'll show you, this was our entry to understand some really exciting molecular insights into the clock. So there's a researcher in Germany who began trying to understand sort of range of human circadian timing. And we call this a chronotype or clock type. Right? Sort of intrinsically, when are you driven to go to bed and wake up? So he's had a questionnaire for about the last 2 decades. I looked it up this morning, and I think it's still searching for a new web home. So I don't know that it's online now. But hundreds of thousands of people have taken this questionnaire, answering really simple questions about when they want to go to bed and when they like to wake up on the days that they work, but more importantly, the days when they have kind of control over their schedule, their free days. Right? And that really dictates what you like to do, right? And so as you look at the sort of distribution of the population, on the left from those that are early chronotypes or morning people to those that are late, the cyan and green bars here in the middle kind of fall under this normative circadian time of about 24 hours. And we know now from human genetic studies over the last 2 decades that there are people who have changes in those clock genes in both the very far left and far right extremes that give rise to very different circadian timing. So I took this quiz a couple years ago, and I'm a so-called moderate morning person. And basically what that means, these blue bars here, I'm one of the lucky 20% of population that wakes up very happily on a weekday without an alarm clock, right? So I'm happy to wake up at 6, on my own. And so the idea in our lab was to utilize knowledge from these so-called early and late chronotypes to really understand more about the molecular basis of timing. Why is it that you can change one aspect of your genome and become a night owl, right? And what underlies that? So ironically, our understanding actually began with this guy. So this is the Syrian hamster. And the reason that we really began to make inroads about understanding the molecular basis of timing was because this has been used to study circadian rhythms for several decades. So here's a picture of Mike Menaker, sort of a very established person in our field, who used this organism. So here's one of his hamsters in a cage. And as you can see, there's a running wheel here. And the easiest way to measure circadian rhythms in an animal is to put them in temporal isolation. So for mice, this is constant darkness. right? And then let them behave, just like Jürgen Aschoff put people in the bunker. The idea was when they're awake, they run. When they're asleep, they don't. So what I'm showing you here on this plot is a whole bunch of days. So this is about more than a month of days. Each row is a day. They're asleep in the dark, and then here's where they're awake. And you can see these thick blue bars are when the mice are running. And what I want to show you is that when you stack this behavior up on a terrestrial day, 24.0 hours, I hope you can see that these little critters wake up and run at almost exactly the same time every morning, near military precision, because internally their clock is like 24.0 hours, right? So their clocks just happen to align up perfectly. So this is really just a lucky blow for Mike Menaker that it became a very easy thing to study when you had something that had a perfect 24-hour rhythm. Didn't require fancy computers and algorithms to figure out periods. So this is really my favorite story in the field. His graduate student, Martin Ralph, noticed one hamster that had a very different behavior. And so it's almost bizarre to look at on this schedule. But what he noticed was that if you imagine all these guys were asleep and then they all woke up at the same time and started running, this one hamster that he named Tau for period, he was awake before the other guys every morning. He was an intrinsic morning lark. And so when you plot that behavior on a 24-hour, hopefully what you can see is that every next day, as you go down to the next line, his behavior advances 4 hours. So he's up 4 hours before his buddies are. And that happens every day sequentially. He's waking up 4 hours earlier because internally he's got a 20-hour clock, but he's living in a 24-hour world, right? So this is the first morning lark, all right? So the identification of this was published in 1988. And again, it took about a decade of really expensive and advanced genetics to identify the gene. And it turned out if we go back to this map of clock genes that we have in our body, that it turns out there's one of these clock genes, which is known as casein kinase 1 delta. This is an enzyme that participates in this pathway. They identified that there was a single change of one amino acid in this protein. And it's the only change that they could see. And when they put that mutation in a mouse, it gave rise to a 20-hour period in the mice, right? So that was published in 1998. And really until then, we haven't really understood, I think, the molecular basis for this. So it's been sort of 2 decades of understanding that we can make a single change in one clock gene and compress the clock from 24 hours down to 20. We now know that there are mutations that are inherited in many of the other clock genes, shown here with red stars, that also give rise to short clocks. So there's not just one way to make a short clock, but in fact many. And in fact, there are also mutations, which I'll talk about one very briefly at the end, in yellow stars here, that can make you a night owl. So as biochemists, this is a treasure trove of information, because now we have specific mutations or variations in these genes that we can ask, how does this happen and give rise to a change in circadian timing? Right, so I'm going to spare you the gory details, but I'll talk a little bit about what we do, and then I'd love to open up for questions. So we began wanting to look at this I would say, the most famous circadian mutant, the tau mutant. And what we did was take this protein, and we inserted this mutation, we made this change. And as biochemists, one of the things that we want to do is think about these proteins as sort of cogs in a molecular clock, right? And so for us, the way that we do that is to see what the cog looks like, to look at the shape and structure of the protein. And how is it that this particular change in one amino acid changes the function of the protein? And so what I'm showing you here, I still am in awe when I look at pictures of protein crystals. But we crystallize our proteins. We make them very, very pure. And just like you have salt crystals in your kitchen, we make crystals of our protein, where every single little unit cell has one molecule of our protein lined up in the same orientation. So this is a number of crystals. You can see that they're kind of thin plates. I don't know if you can see the scale bar here. It says 200 microns. Each of these crystals is smaller than the width of a human hair. Right? So they're very, very tiny. We have to work under a microscope. And what we do is we grow these crystals, if we're lucky. We fish them out with little teeny fishing loops that are about the width of a human hair. And we put them in front of a big X-ray source. So in this case, we go to the Berkeley National Lab up in Berkeley. And we put our crystals in front of the X-ray beam and we shoot them. And the structure of the protein inside those crystals diffracts out the X-rays, And we give rise to these very beautiful patterns, and these regular patterns, right, that arise because there's a physical entity within the crystal that's bouncing those X-rays around. We can then take these patterns, and with some very fancy computational work, we can build backward to see what was the shape or density of the molecule within that crystal that gave rise to that diffraction pattern. And this is sort of a mesh of a network, and it looks like a big glob right now. And what we can do is fit our protein structure into there. And so I fit sort of the actual bare bones of this protein molecule. This is the tau mutant. I'm going to take away that mesh. This is still a nightmare to look at. As biochemists, we don't look at protein structures in sort of a chemical way because unlike chemists who work on beautiful small molecules, our molecules are very big. It's very hard to process this information. So we tend to depict them with what we call a cartoon mode. And this kind of mode tells us about the shape of the chain of the protein and how it looks. And understanding this is kind of like, the best analogy I can make is if you want to understand how your car works, you can open up the hood and ask, like, for example, what a spark plug does, right? A biochemist will open up that hood, pull that plug out, see what it does, see what it looks like, see how it fits into the car. And that's what we're doing when we look at protein structure. We're really trying to understand at a fundamental molecular way what this does, right? So I'll tell you, we have two structures now of a normal, sort of wild type, healthy CK1 delta, and this tau variant shown here in gray, and I've stacked them right onto one another. And hopefully you can see in many places of the protein, you can't distinguish the two molecules. They're identical. But hopefully you can see this little movie is kind of morphing between the two structures, and it's wiggling and jiggling. And so we know that change of one amino acid causes this protein to kind of flip a molecular switch. So for example, the single amino acid change going from an arginine at position 178 occurs right here by this negatively charged group that binds onto the kinase. And we've been very fascinated and excited in our lab now to understand the basis for this little flicker of this switch. I will tell you, this little difference in one protein has enough power to change your clock from 24 hours down to 20. Right? And so we've been trying to get to the basis of how this happens, and once we understand that, can we regulate this process? Right? What's exciting to us, I'll go back a sec, is that hopefully you can see there are other parts of the molecule that are wiggling and jiggling a bit. And in fact, we can map 3 other mutations that also give rise to a short circadian period to parts of this protein that are wiggling and jiggling in concert together. And so I'm not going to bore you with too much of the detail, but I have a fantastic graduate student who has a really phenomenal body of work now that has pinpointed exactly how this mutation gives rise to a change in circadian rhythms. And it's really an exciting thing to put together. And so if I bring it back to this map of all of these core clock genes, this is but one example of how we can bridge the gap between understanding how circadian rhythms control behavior and physiology down to the molecular basis by which timing is measured in the human body, right? So I've showed you one example of a morning lark here, which sort of puts us over on this left side of the curve. And I'll just take 1 second to give you one more slide about a night owl, and then I'll open for questions, because I think this is perhaps the most exciting and translatable field finding. So I'm going to show you some human data. So this tau mutation, although exciting, originated in hamsters. Now, some of the other mutations in CK1 that we know about are also found in humans, but they're quite rare. So these particular morning lark variants are maybe 1 in 100,000 people. So not incredibly prevalent. But one of the Nobel laureates, Michael Young, published a paper 2 years ago identifying a very prevalent human variation in one of those clock genes called Cry1. And by prevalent, I mean 1 in 75 people of European descent. And so I don't know how you roll with understanding genetic polymorphisms, but 1 in 75 is incredibly prevalent. So there's probably 1 or 2 people in this room that have one of these variants that have passed it on to their children that have an inherent basis towards sort of night owl behavior. And I want to show you the consequences of that. And so if we put sort of a fancy circadian rhythm activity monitor on humans, no, I'm not going to put you in a running wheel and ask you to run, but we can put sort of a very fancy Fitbit that measures activity as well as measuring exposure to light, right? So if we do that on a control subject that has a 24-hour rhythm, the little red triangles are indicating when they went into bed. So this was completely elective timing. And then the blue circles indicate when they get out of bed. And so hopefully what you can see is that they're active during the day. So just like mice, activity is measured by those little black bars, right? So they're active during the day, activity kind of slows down at night, and they get into bed right around midnight almost every single day by choice. They sleep there around 8 hours or so, and then they get out of bed. This is a typical circadian profile of someone who has a healthy, sort of robust 24-hour rhythm. Now, you don't have to be a sleep study researcher to realize that if you have this variation, your clock is completely messed up. I know that's not a very scientific way of saying it, but you can look at the timing for sleep onset often extends well past midnight here, like to 3 in the morning or 4 in the morning. Sleep bouts at night are often limited to only 3 hours at minimum. And importantly, there's a whole lot of daytime napping, because what do you do if you don't get enough sleep at night? You fall asleep during the day, right? And so what was amazing was Mike Young, who was one of the people who identified this first period gene. He identified and tracked down the genetic variation that gives rise to this. And it was one change in this Cry1 gene that basically was responsible for taking a 24-hour clock and making it a 25-hour clock. So just a 1-hour change in internal timing is sufficient to delay melatonin and mess up your sleep onset timing. And I'll tell you, I have another graduate student in the lab who's approaching a structural sort of consequence of that. This is another representation of a structure of how Cry1 binds to our transcriptional activators, clock and BMAL, that we solved in the lab several years ago. And basically, what Mike Young found was that there's a little teeny part of the protein in a very flexible region of the protein, that this is the region that gets deleted if you have this night owl syndrome. And work from my student, Giancarlo Perico, which we're getting ready to publish, has shown that this region actually acts to control how these two proteins come together, right? And so what's truly exciting for me is to be able to bridge the gap from human behavior and physiology down to the molecular nitty-gritty of how timekeeping arises, right? So I've framed the discussion today in terms of kind of when you like to go to bed, which is, I think, a cute way of thinking about one of the consequences of circadian rhythms. But I want to close by just pointing out that nearly every aspect of your physiology and behavior is under circadian control. So everything from when you like to go to bed, right, your sleep-wake cycle, to your appetite, your cognitive performance. In terms of physiology, we know that there are amazing consequences metabolically, that you are not the same person at 5 a.m. as you are at 5 p.m., right? In terms of how you metabolize food, what you do with those nutrients that you eat. And I can address that later if anybody has a question. Your ability to produce hormones, control cardiovascular function, your ability to respond to immune challenge is under circadian control. And likely, even your ability of your cells to make decisions on whether they should divide or not, right? So this just brings me to this sort of take a step back, that throughout evolution, life has found a way to use the temporal organization given to us by Earth's rotation to neatly control and systematically organize our physiology. And this has really been, I would say, under assault by industrialization, right? And I'll just close by telling you, and reminding you that light is a drug for your system, and light at night is telling your body that it's daytime, right? And so we just now know that there are pervasive consequences to shift work and light at night, to the point where World Health Organization has classified shift work as a probable carcinogen, right? Because what it does is give you permanent jet lag. It releases those daily breaks that get put on cell division, and as a consequence, we know that there's enhanced risk of breast cancer and other forms of cancer that come with that. So if there's anything I've hoped to convey to you, it's that circadian rhythms have a really powerful and beautiful way of coordinating your physiology, and that really intricately tie us to the planet, right? So I'll just close by thanking, it's been a fantastic opportunity to work at UC Santa Cruz. This is just some photos of people who are currently in my lab and collaborators at the Duke NUS Medical School in Singapore, where we've been working on this Tau project, as well as some friends of mine at UCSF, where we're working on pharmacological approaches to manipulate circadian timing and resetting for jet lag. And if there's time for questions, I would love to entertain them. Thank you for your attention. Up here in front.
I'd be interested in your recommendations for enhancing health. I do have blackened curtains in my room, but be interested in.
Yeah, so what we understand generally is that I showed you those oscillations that happen at the gene level. That the taller those amplitude, the taller those oscillations are, the stronger those time cues are sent to your body, right? So basically, the better that circadian control is communicated. And we know that throughout aging, the strength of that clock tends to diminish, and those time cues aren't conveyed as well. So one of the things that we think can really help enhance overall health and well-being is by enhancing circadian function and keeping everything running. So the way that we recommend to do that now is with light. So you mentioned that you have blackout curtains in your room. That's fantastic because that reduces light exposure at night. But there's one other aspect, and it's like as if this were happening on cue, that we need light exposure during the day. So most of us spend our adult lives inside rooms like this that are nearly 1,000-fold times less bright than natural sunlight. And so we're starved for sunlight during the day, and we get too much light at night. And so what we know from many other studies is that if you get out during the day for at least a half hour or an hour, and you take in bright sunlight, it has a really profound effect in helping to consolidate and organize your circadian rhythm at the molecular level. So people like Ken Wright at the University of Colorado at Boulder has shown this in a research study by taking people camping. And it's a really beautiful study where he was able to fix and consolidate circadian rhythms by getting people out in the day and then reducing light exposure at night. Right, so for right now, that's our best hope. I will tell you I have friends who are working on pharmacological strategies to enhance circadian rhythms, and the results are very promising. That there are some natural products, including some things that come in tangerine peels, that can help to stimulate and enhance circadian rhythms. And the work, I'd be happy to share a paper if anybody wants one. The consequences of that are that it works to fight off all of the bad things that come with disrupted circadian rhythms. So better health, better metabolism, better sleep.
Is this bad to wear in the daytime?
Well, they do block a lot of light, right? So, I mean, you need photons into your brain. You need light information to your circadian rhythms to tell people, tell your master clock that it's daytime. Yeah. In the back there was one?
Because the sun goes around, the Earth goes around the sun in an ellipse, then the sun gets out of kilter with clock time by up to 20 minutes or so, in kind of way. That's shown by, at least in Europe, by the Anna level, which occurs on church sundials and in graveyards and all the rest of it. Has anyone ever taken these people with whom you can tell a very accurate, as it were, 2 a.m. Working time, and checked whether around the time of maximum extent one way or maximum extent the other way, it actually shifts with respect to the clock time?
So you're asking, I guess I'm trying to find out what you're asking. You're asking whether people who have sort of night owl-like behavior shift back depending on where they are geographically?
No, not geographically. Well,
or like seasonally, I guess. So, I mean, certainly circadian rhythms, the sort of internal measure of...
Well, if it's the light that's resetting everything, then it too should shift by the amount of time shown on the other level between the times of maximum lateness of the sun and maximum earliness of the sun with respect to clock time.
So this is an amazing question, but this is where there's a kind of, I think, a divide between the way that most of this research has been done in the lab setting versus this, So in a lab experiment, where people are studying circadian rhythms, they usually do it in a perfect day, 12 hours of light and 12 hours of dark. And so this is a great question. People have looked at the duration of light and resetting, but very few people have done what I would say is a very elegant and nuanced experiment that you're suggesting. There are people now beginning to do this kind of work as we have better sort of approaches to monitor behavior out in the wild. So I have a colleague at Utrecht who's looking at the effect of being out and looking at changes in day length and entrainment, but that work is basically ongoing just now. So it's what you've described as a very elegant experiment, and no one's done that yet. Yeah. Yeah, up here.
Could you talk a little bit about melatonin and what taking melatonin might do either to reset and also to help being on the clock and and negative sides of melatonin?
Yeah, so I'm not, I mean, some of those things I can't address, I think, with a lot of knowledge, but I will tell you that melatonin doesn't reset your clock. What it does is help you sleep. So if you fly to Europe, right, and as I'm going to do in about a month to go speak about some of our work, when you first get there, you're often not tired at night because you're 9 hours back. So if you pop a melatonin, what it does is bring on this kind of hazy, sleepy feeling that allows you to fall asleep when normally your circadian system is priming you to be alert. What it doesn't do is affect that molecular clock I've shown you, right? And so what it does is sort of help prepare you and adjust you, and really it's a practical benefit that you need sleep in order to feel good the next day. So to the extent that melatonin affects your clock, it doesn't. What it does is allow you to sleep and wake up. The negative consequences I know of are that typically when you take a melatonin pill, you might be taking 5 or 10 milligrams, which is like 1,000-fold over what might be in your body. And so some people get what's known as the melatonin hangover, where they might take too much or take it too late, and when they wake up the next morning, they feel kind of groggy, right? And so that's why many people stop taking it, because they don't like that feeling. And so part of it is knowing when to take melatonin, and ideally melatonin levels are supposed to come up as the light dims, right? So bright light will suppress your internal melatonin levels. And so if you get to, say, Rome, I don't know why that's my favorite example, but if you get to Rome and it's 11 p.m. local time and you take it, that's a bit after the sun already went down. And so it's kind of a little bit late at that point to take it. But it will help you get to sleep that night, and you'll feel more like a human the next morning if you do, right? Yeah. Does that address your question? Okay. Yeah.
I wanted to ask about when I fly to Southeast Asia, I have about a 14-hour time change. And I always feel like it's easier to adjust over there than it is when I come back.
Yeah.
Yes. Is that what science shows?
So absolutely. And so this is actually a fascinating area. So what happens when you travel? So imagine that sinusoidal wave is that you don't change the shape of that wave, but you take that graph and you shift it forward or you shift it back with light. So we call this a phase shift.
Yeah.
So it turns out that advancing your clock, which is what we have to do when we fly east, is hard. And usually delaying your clock is easy. Now, why that is at the molecular level, I mean, that happens at the molecular level, right? And that, I think, manifests in terms of behavioral responses. It's not known, but I will tell you one study that Alec Davidson published probably a decade ago. He took a series of sort of aged mice, and he basically did the molecular equivalent of flying them to Europe or to Hawaii. So a 6-hour sort of shift in light, either back or forward. And the mice that he shifted towards Asia, we'll say, that went that direction, they were just fine. They had normal sort of locomotor behavior. They were just fine. The mice that he flew to the east started dying, right? And so, sorry, you can still go to Europe, I promise. But what he found, there are physiological consequences that we don't yet understand about phase advances that we're trying to really tease out at the molecular level currently in the lab. So what you're reporting phenomenologically, we know happens at the molecular level and at the behavioral level as well. So that's a really common phenomenon that it's much easier to delay than advance. And I will tell you, there's something interesting and molecular about it, and I have 2 postdocs in the lab who are working to understand what is the basis by which our clock gets moved forward and backward. We don't understand that yet, but I'll tell you is that we know it involves that period protein. So that is the central sort of state variable of the system that defines timing in the clock. So I think there's still many more interesting discoveries to be made. So maybe one day I'll have a real answer for you. Yeah.
And are you saying that if one were to study a person who lives in Seattle all their lives, in contrast to one who lives in, say, Palm Springs, would perhaps one group be more susceptible to disease or something of that nature than the other?
Well, yeah, absolutely. So for example, you've probably heard of seasonal affective disorder, or SAD, right? So I actually grew up in Seattle, so I can speak to this authoritatively. There's a high incidence of seasonal affective disorder. In that case, because we have short days in the summer, we actually only get sunlight in the summer. And so, absolutely. So we know that kind of at a level of seasonal affective disorder. The epidemiological studies now to look at other diseases that we know now accrue with circadian disruption are ongoing. I would say at the human level, we don't have a complete answer to that yet, but we know from, say, studies of Drosophila melanogaster out in the wild that we can see changes in clocks that have evolved as a function of where you are, if you're close to the equator or if you're up in northern Europe. So we know that animals do adapt ecologically to their niches. So there might be, exciting things yet to come on that front.
Okay, maybe one more.
Okay, one more in the back here.
So if you're a night owl, and you said being a morning lark, that 10 to 12 is an optimal time, but for a night owl, what would be the optimal?
Fantastic question. Yeah, all those values I gave you in the beginning were for a standard sort of 24-hour circadian clock. So if you were a night owl and you were following your own internal clock, right, you were going to bed when you wanted and waking up, say, later than other people, then that would be shifted back, that ideal timing. And it typically happens about 4 hours after waking is your ideal cognitive performance. So if you're a night owl and you're waking up at 10 a.m., right, if you were sleeping in, then that would happen at 2 a.m. So it really does follow sort of your internal circadian timing.
But your daytime at your most productive would be like 2 to 6.
Yeah. And I can just tell you anecdotally, for my postdoctoral studies, I had to leave the field of circadian rhythms because I had to pick up some tools, how to do all this molecular stuff, right? No one was really doing that. And I had a colleague, I'm a morning lark, right? I had a colleague who was a total hardcore night owl. He would write grants after midnight. He would go for runs. I mean, at midnight, I'm good for nothing intellectually, right? Like I can maybe watch a movie with my kids if they're still up, but that's it. And he would have his most intellectually fruitful times between like 9 p.m. and 1 in the morning. And so, if you're a morning lark or a night owl, we all have preferred times of day. So this is just another extent to which the circadian process is controlling how you interact with the world, over the course of the day.
Okay.
Great, thank you.
So, I think you can all see why Carrie is one of our rising stars on campus. And I should mention that another achievement of hers is to get funding from the Howard Hughes Foundation, which for a biologist is nirvana. Right. So if you have questions for her, you can come up and talk to Carrie. So thank you, everybody, for coming. This wraps it up for the year. We will see everybody in September.